Good morning, everyone. Before we begin, I would like you all to please stand up and attain 2 minutes of silence in the memories of victims of Pahalgam attack. Thank you, everyone. It is with deep pride and great reverence that I, Shweta Sharma, welcome you all to the 8th Professor Kaul Memorial Lecture here at CSIR National Botanical Research Institute. This annual Memorial Lecture is a moment for reflection, inspiration, and dialogue. Each year, we invite a distinguished speaker who embodies the same innovation and impact that Professor Kaul represented. We are incredibly fortunate to have such distinguished guests with us today. Please join me in welcoming our chief guest, Padma Bhushan Professor P. Balaram, former director at IISc Bangalore. Along with him, I invite Dr. Ajit Kumar Shashne, Director CSIR-NBRI, and Dr. Vidhu A. Sane, Chief Scientist and Coordinator ACSIR, to grace us with their presence on the dais. Please join me in welcoming them. I would request Director Sir to welcome our chief guest with a bouquet of flowers. Lighting the lamp signifies the dispelling of darkness and the welcoming of knowledge, wisdom, and enlightenment. It is a gesture that not only honors our rich cultural heritage, but also symbolizes the illumination of minds in the field of scientific research. I would now like to invite our distinguished dignitaries to kindly come forward to light the lamp. And I also request Poshali to please recite the Saraswati Vandana.
या कुन्देन्दु तुषार हार धवला या शुभ्रवस्त्राभृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवै सदा वन्दिता सा मां पातु सरस्वती भगवती निशेष जाड़ा पहाड़।
I now request our guests to please pay a floral tribute to the memory of Professor K.N. Kaul. To give us the welcome address, I now request our director, sir, to please come on the dais.
A very good morning to everyone. Respected Padma Bhushan, Dr. Professor P. Balaram sir, in front of me, Professor Ram Rajashekharan, Dr. Dhawan, my colleagues, student friends, and all the staffs of NBRI. Today is a great day that we are in memory of Professor Kaul, the founder of this institute. We are celebrating in memory of him. And we have the distinguished guest, as told by my friend here. It is very difficult to get him. He is always busy. And he has several scientific work, and you cannot get a better person than him. He is not known only in India, but he is a scientist known all over the world. Thank you, sir, for accepting our request and coming here and blessing us. Also blessing the students, mainly. Our students may be 200 or so. Also the students from other institutes of CSIR and some of the universities nearby, they are participating in this student science festival, plant science festival. I think, I'm sure they will be greatly benefited by your wisdom, and they will be definitely inspired by your presence. Thank you, sir, for coming. Also, I welcome Professor Ram Rajashekharan here and Dr. Alok Dhawan. I think he will be staying for the whole time with us. Also, I welcome my colleagues and all the students for whom the festival is, by the student, for the student, of the student. So they have designed all the activities themselves, and they are working very hard in NBRI. They will be definitely inspired. I welcome you all. Sir, this is the Institute of Historical Significance, and today morning we have seen a banyan tree. So this is related to the first war of independence, where 32 soldiers, British soldiers, were being killed by the great Uda Devi. The statue is there in the square there when we come to NBRI. When we go to the other compound, and this compound in between, the statue is there with a gun in her hand. Later on, she has been searched out and killed by the Britishers. So this is the historical place, and the importance of this place is related to botany, environment, and we have the departments of taxonomy, starting from taxonomy to molecular biology, genome editing. So we identify plants, and the plants which are actually not been taken up by the mainstream. So these plants we do research on, in addition to the gaps in the mainstream plants: biodiversity, environment, microbiology, agronomy, soil science, breeding, everything, all aspects of plant science is being worked upon here. So this is an institute, and the students, they are eager to listen to you, so I will not stand in between. Thank you very much again, all of you coming here. I welcome you, all of you again. Jai Hind, Jai Bharat. Namaskar.
Thank you, sir. I now invite Dr. Vidhu A. Sane to the dais to please enlighten us with the legacy of Professor Kaul and to please introduce us to the chief guest of the day.
Thank you, Shweta. Namaskar. Suprabhat. I welcome you heartily on this 8th memorial lecture in the memory of K.N. Kaul. So it is a rare occasion because we are organizing this lecture in the memory of Padmanabhan, Kaul, and we have a guest speaker who is also a Padmanabhan. So a round of applause. So we who are at NBRI owe to Professor Kaul a lot because he was the founder of NBRI, then known as NBG, National Botanical Gardens, Botanic Gardens, in 1948. He was a botanist, horticulturist, he was a plant collector, he was herbarium, he had devised a fern, he discovered a fern, and so a fern is named after him. Professor Kaul was, in true sense, a interdisciplinarian. We talk about this interdisciplinary science nowadays, but he was, in true sense, doing all the things. So he was the one. We have a lot of land which is alkaline, not usable. So he was the one who proposed a reclamation of soil and land. And so we have a big land in Banthra where we now have a research station. So we owe a lot to Professor Kaul. Now, it is my privilege to introduce our guest. It is really difficult because he is a renowned scientist in the world, not in India, but in the world. He is Padma Shri, Padma Bhushan. So he did his master's from IIT Kanpur, nearby city of Lucknow. He had done his BSc graduation in Fergusson College, Pune. And then he went for his PhD degree, doctoral degree, and that he got received in Pittsburgh, Pennsylvania. He worked with the Nobel laureate, Dr. Vanderwood. So he is a phenomenal person, and I'm sure that Professor Balaram will talk something about him. And there he worked in the synthesis related to synthesis or understanding the synthesis of erythromycin. And I think Dr. Vanderwood has done a lot of good work, and Dr. Balaram is taking that legacy very well. So Dr. Balaram is a recipient of number of awards, and I don't think that I am really capable of talking about those awards, but a few of them is TWAS award, which he received, and then the latest one, which he received in 2021. This was conferred by American Society, one of the societies. So this is also very important award which he got. So many of you, I don't know, but many of you, I am sure, have listened to him. In YouTube, he has many lectures, and when you will hear him, you will see what a flow, what an information. It's beautiful. So I am not coming in between you and Professor Balaram, and just go into the flow of Professor Balaram's wisdom and everything. Thank you very much, sir. I forgot to tell you that he is, for all ACSIR students, he is Chancellor ACSIR also. So this is very important for everyone. So in NBRI, we have Dean Biological Science also, Dr. A.P. Sane, and now we have Chancellor with us. So he and you, please, sir. So do you got this mic? This is
Should I put this on?
if you want to move around, otherwise.
Maybe I don't know what I'll do.
Let me switch it on. Sir. Let me switch it on. It's not on. Sir.
It's not on.
It's not on.
the slides come on. You don't put the screen down? I think maybe that's better, isn't it? If you put the screen down. Oh, if you move the projector down, fine, fine, thank you. There's so much technology nowadays that you don't know who you're talking to. And, you know, I'd like to thank the director, Dr. Shasni, for inviting me to be with you today at NBRI. It's a real privilege to come here to give a lecture in memory of Professor Kaul. And going around NBRI, of course, I realized that Dr. Kaul's legacy lives on. And the subject of my presentation itself is something that I have been thinking about in my retirement years. And the one thing that you would have guessed by the fact that Vitu introduced me as the Chancellor of CSIR is that I must have reached a fairly advanced stage to be simply signing on degrees. And that's one of the jobs that I do. What I'd like to talk about is evolution, and I'd like to talk about evolution in its most general sense. Evolution is the word which really is associated with Darwin, it's a word which is associated with biology, but it turns out that evolution is a word which can be used in the widest possible context. So when I was told that you're celebrating a plant festival, plants are of course only one part of biology, and there are other parts of biology which are also vast, and there's a common thread which runs through all of biology. That common thread is chemistry. The biochemistry is the same, the genes are the same, you don't have a different genetic code in one organism or the other, you have the same genetic code. So everything in biology can actually be traced back to a common ancestor. And the question that you might ask is, what is the last universal common ancestor from whom we have all descended? That would of course be some unicellular organism. So in my starting slide, what I really have are the two foundational pillars of the two subjects which I will talk about, chemistry and biology. There's Darwin on the right and Mendeleev on the left, and you will find that the periodic table and Darwin's ideas of natural selection underpin almost all our current ideas of chemistry and biology. Ironically, these are the two subjects which were removed from the 10th class syllabus by the NCERT a couple of years ago when they were trying to reduce the workload on students. In the middle, I have a quotation from Arthur Kornberg, the biochemist who wrote most thoughtfully in the latter part of the 20th century about what molecular biology has really done to biochemistry. And Kornberg lamented the fact that molecular biologists and biologists in general pay very little attention to chemistry. And then he called chemistry in that article the lingua franca of the medical and the biological sciences. So you can't do anything in medicine or in biology if you don't have a reasonably sound understanding of the underlying chemistry. Does this click on? Okay, fine. Now what we are resting on in 2025, and I think I have, it's 11, approaching 11:10 now. When should I stop? We do. I'll stop, okay. So I don't want to delay you, we're already a bit late. We are dealing with the background that we've learned in a century of science in the 21st century. I am pretty much a child of the 20th century, and most of you who are in the audience are children of the 21st century. And if you look at a century of science, you can ask, what did the 20th century begin with? The 20th century really began with the quantum, and the quantum was a word which was introduced into science by Max Planck and Albert Einstein. And it turns out that we began with the quantum, and in 1925, which is exactly 100 years ago, this year is the centenary, Werner Heisenberg wrote his famous paper, which is the starting point of quantum mechanics. Why am I talking about physics over here? The only reason for this is quotation which comes from Paul Dirac a little bit later at the very height of the quantum revolution. What he said was the underlying physical laws necessary for the mathematical theory of a large part of physics and the whole of chemistry are completely known. I have highlighted the whole of chemistry. So, quantum mechanics really allows you to understand and make the most remarkable predictions about the structure, behavior and chemistry of almost any molecule. Therefore, it underpins all of chemistry. Subsequently, after the 1950s, post 1953, when Watson and Crick published their famous paper on the double helix of DNA, Oswald Avery discovered DNA as a genetic material in 1943. So, we have had about the last 50 years of the 20th century dominated by what you call biotechnology. And the moment you call it biotechnology, you immediately imply that there are some useful things which are going to come out from the study of biology. And sometimes the idea that you are going to always be doing something useful is not entirely supportive of scientific activity. Because if you are doing something always useful, you are not likely to be able to find something that you do not understand and you have suddenly understood it. And maybe this will be useful much later. At the beginning of the 20th century, if anyone had asked, would the quantum have any use? Nobody would have said that it's of use to anybody. But today, of course, you will find that the government of India now is launching a mission which they call the quantum mission. So, the quantum mission now uses a word which is 125 years old, and nothing was anticipated at that time. The year 2000, the century ended with that picture there. That is Craig Venter and Francis Collins shaking hands when President Clinton announced the completion of the human genome sequencing project. That is 25 years ago. You can of course ask a question, how much have we learnt about human beings after the sequencing of the human genome? How many advances have taken place after human genome sequencing became commonplace? But in thinking about biology, we have to ask a question, and that question is, what is life? It's a question which has been asked by many people before, and it turns out that there are many views of life. The best one is due to Darwin in his book, The Origin of Species, where he talks about selection, adaptation and evolution, and he says it's so wonderful that from so simple a beginning, what is that beginning? From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. So, if you go to one of the greenhouses in your garden over here and you see all the plants which are there, you just wonder. But if you go to any other place, if you go on a safari, you will again look at all the animals and you will wonder. And sometimes you do not have to go very far, you only have to look around you and you will wonder at the diversity of even organisms which are a single biological species. The definition of life, of course, better given by Jacques Monod, the molecular biologist. He said there are living systems, there is no living matter. Chemistry deals with the inanimate, biology deals with the animate. So, biology deals with living matter, chemistry deals with non-living matter, but matter is matter. And physics has provided us our understanding of the constitution of matter. It is really matter that has always been the subject of science right from its very beginning. And he says no substance, no single molecule possesses all the paradoxical properties of life. And he says they are present in living system nowhere below the level of a cell. So, the cell then is the unit that we have to worry about when we are talking about the conversion of chemistry into biology. Now, cells are to biology what atoms are to chemistry. So, when you talk about chemistry, you always talk about atoms, you talk about molecules, and the definition of atoms or the importance of atoms, you can't do it better than the physicist Richard Feynman. Feynman, in his famous lectures on physics at Caltech in the 1960s, this is volume 1, chapter 1, he defines matter is made up of atoms. And then he asks a question, a very interesting question, and the student should ask this question: if in some cataclysm all of scientific knowledge were to be destroyed and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? You can ask yourself the question, why is Feynman thinking of a cataclysm in which everything is destroyed? Because he was teaching physics in the 1960s. And in the 1960s was the height of the Cold War between the Soviet Union and the United States. And every day one feared that there would be an outbreak of a nuclear war. Now, it turns out that 60 to 70 years later, nothing's changed. We also worry even today that there will be an outbreak of a nuclear war at any instant of time. There has never been a problem which has been solved in the case of human conflict. And so one must worry. Suppose everything is destroyed, how do you start all over again, building up the edifice of science? And Feynman answers this question. He says, I believe it is the atomic hypothesis or the atomic fact, whatever you call it, that all things are made up of atoms or little particles. We now know what atoms are. We know what atoms are made of. We know how atoms combine together to give molecules. That is the edifice of chemistry. Why am I thinking about all of these? That's largely because of something that happened over the last few years. In 2020, when the coronavirus pandemic came upon us, hardly anybody knew what a coronavirus was. Now, of course, everybody knows what a coronavirus is. And at that time, even virologists did not know what the coronavirus was because it was an obscure virus. It hasn't really caused too much of a problem for human beings. And suddenly it was there. It shut the world down. It spread throughout. I don't think there's a single person in this audience who has not been infected. Even if you've not been sick, you've been infected. And therefore, you have this little particle, 90 nanometers in diameter. And this particle gets into you, multiplies and does this. That's biology. A virus gets into a host cell, uses the host machinery to replicate, and then there it is all over. What is this plot here? The inset is the number of deaths, the number of infections, etc., which you would have seen. You can see how many people died at what point in time during the coronavirus pandemic. The death toll now is probably close to 7 to 8 million people all over the world, and that might be an underestimate. But over this period of time from 2020 to 2024, where this graph has been made, one protein on the surface of the virus, those red spikes there, has been sequenced, and the mutations which it has accumulated have been plotted. These are the so-called variants which came through the pandemic, the alpha variant, the delta variant. At different points in time, the newspapers will tell you that you had variants. So what was the virus doing here? The virus was evolving. What was it evolving? Darwin has told us that there must be a selection pressure. What was that pressure? That pressure is, of course, what the human immune system is mounting. Is the pressure mounted by the human system the same everywhere? No. It is different in a South Asian population. It's different in an African population. It's different in a Caucasian population. It's different in a vaccinated population. It's different in an unvaccinated population. So under these different selection pressures, mutations have evolved. And eventually it turns out, but by 2023, the middle of 2023, the virus had accumulated as many as 60 mutations. Those numbers that you see there, right at the beginning, one mutation drove the pandemic. Afterwards, 10 mutations killed so many people. But by the time you come to 60 mutations, evolution has worked. The virus has no intention to kill the host. This is one of the interesting things in biology. Biology has no purpose. We invest it with a purpose. In biology and organisms, major imperatives are survival and reproduction. And survival only until reproduction is over. After that, there is no particular advantage to survival. If, as I was interested in proteins and the structure of proteins and so on, a great deal was learned about membrane fusion. It turns out that this coronavirus spike protein is the only one for which a structure is now available in what is called a post-fusion state. So, for the first time we are able to understand in some kind of atomic and molecular detail the processes of fusion. But that is not what I want to talk to you about today. To ask a question, the virus, all of you know the virus was around, virus causes a great deal of problems. Is the virus a living organism? And the answer of course would be, the answer would depend on who you ask. Some virologists like to classify viruses as living organisms, others do not like to classify them as living organisms. And if you look at a biology textbook, you will find viruses do not find a place in the tree of life. You have bacteria, eubacteria, archaea and eukarya, but you do not have viruses. So, if viruses are not a part of biology, then viruses must be a part of chemistry. And you will of course say, yes, viruses have no existence on their own, or they need a host cell to multiply. And I would say fine, so shall we call a virus a parasite? You will think about it a little bit and say, yes, malaria for example, the parasite, we call it a parasite, it either lives in the mosquito or it lives in a human being or in any other animal, and therefore it just moves from one host to the other. It has two hosts in its life cycle. This virus has only one host in its life cycle, so it is also a parasite. Then of course I would ask the question, are all of you now free living organisms? And you imagine that you are free living organisms. The real fact of the matter is you are not. And the real fact of the matter is you are not, because for example, biochemistry teaches you that you have lost the ability to make so many amino acids, and therefore you will classify amino acids into essential and non-essential amino acids, and you must get the essential amino acids from your diet. You must get so many things from your diet, so you can't live without the outside world. So you are also a parasite. So everything is a parasite in biology, and there are very few organisms which actually do not need other organisms for survival, at least in the most elementary stage. Bacteria sometimes do not need anything else to survive, and therefore we can ask, life must have originated as bacteria. You might ask about plants, and I will come to that in a moment. Plants are a little bit more sophisticated, and plants are actually quite complicated, and one has great difficulty philosophically when you deal with plants. We sometimes do not consider plants as living organisms, because those who eat plants are vegetarians, and those who eat animals are non-vegetarians. So this would automatically tell you that somehow your classification of a living organism has got mutated in that definition. But biology is a subject which is fascinating for another reason. Biology, if you remove human beings from the equation in biology, then biology is completely sustainable. Look at the processes that I have listed on this slide. These are processes which you would see in 10th class, and they are at the core of biology. But it turns out they are the only processes that you think you have understood, but you have not, because nobody has understood them. And the details are still a matter of discussion. You have two things here. One is photosynthesis, and the other one is respiration. So if you take plants and animals, it turns out of course that plants use the energy of sunlight, and since they use the energy of sunlight to fix carbon, they produce carbohydrates. And then of course the animals eat the carbohydrates, burn the carbon and produce carbon dioxide. And so animals put out carbon dioxide into the atmosphere, and in the process of putting carbon dioxide out into the atmosphere, they use up oxygen which is needed. Oxygen is used in respiration in a very tangential way. Oxidation is used, oxygen is used as the terminal electron acceptor in the mitochondrial electron transport chain. That's all. Before oxygen came on Earth, other molecules were used as the terminal electron acceptor. Microbiologists know this very well. After all, there are organisms which are anaerobic. They don't need oxygen. They are still respiring. So evolution asks questions, how did all of this come about? How did things become so incredibly complicated? This sustainability you can see here. You can see in one direction of photosynthesis, we are splitting water into hydrogen and oxygen, except that nature hides the hydrogen. In the other direction, we are taking oxygen and converting it into water. That's only an incidental reaction. Today, of course, what the government would like to do is to take water and produce hydrogen from it, because then you will have a new kind of, you can get rid of carbon burning fuels and you will have hydrogen, which you would call a clean fuel. But the one thing you must remember, there is nothing clean about any technology that you do. All technologies use some process or the other, which is now environment damaging technology. Biochemistry tells us that the two enzymes whose structures I have shown there, they are membrane proteins, complex membrane proteins doing the simplest of reactions. Water splitting is simple in concept, very difficult to do in practice. And you need all these structures, but these structures have evolved over time. How did nature invent this biochemistry? And that really is the question that should bother you. So here's the timetable of evolution. In science, it's useful to think on a grand scale, and it's also useful to think on a grand time scale. If you think on a grand time scale, you realize how insignificant anything that you're doing is. And it turns out that your entire lifetime will not even be a dot on this time scale, because we start 5.67 billion years ago. A billion is 10 to the 9th. When I was young, we did not know how many zeros there were in a billion. Today, of course, we know, because of all the scams that we've had, everybody is used to any number of zeros in financial figures. So 567 million years ago, that's when the Earth was born. So everything that happens before the Earth is born, let's dismiss it as physics. That's when the universe is born. We require the Earth and the solar system to form before we can start thinking about life. Now on Earth, there is no life to begin with. It's not a planet on which anything can be there. Things have to slowly evolve. What is that period of evolution? When did the first archaebacterium, for example, come on Earth? The estimated times for these are somewhere about 3.5 billion years ago. So you can see for about 2 billion years, what has evolved? What has evolved, I will tell you there, is geology, chemistry, and also the elements of chemistry that are needed for biochemistry. Oxygen evolved only after almost 3 billion years. 2.5 billion years ago is when the grand oxygenation event is pictured. Today there are fairly accurate estimates, including a very recent paper which appeared in Science 2 or 3 weeks ago, which places that O2 event somewhere there. That's when cyanobacteria invented photosynthesis. After cyanobacteria invented photosynthesis and the Earth's atmosphere became oxygenated, you then have two kinds of biochemistry. Biochemistry before oxygen and biochemistry after oxygen. There are many, many enzymes in biochemistry which use molecular oxygen as the substrate. None of those were present in the earlier timescales. And when did the first animals come about? They come about around 500 million years ago. 5 billion years have passed before the first worm appears. And since you are in the National Botanical Research Institute, I might tell you that plants are later. So if you look at the subjects that you study, I would study physics first, geology next, chemistry afterwards, biochemistry, microbiology, zoology, and then only come to botany. And it's only when I go in the sequence would I really understand the complexities of what we are dealing with. By the time we come to medicine, it's almost not there. Engineering was not a word which was in common usage in the 19th century. So engineering is a 20th century word. And so everybody wanting to do engineering and medicine must know that engineering and medicine rest on human evolution, and they rest on all these many billion years worth of biological evolution. So here are life's imperatives: survival, reproduction. What are the ingredients for life? Information, energy. Information we all know: DNA, DNA, DNA. Everybody knows DNA. And DNA contains all the information that's necessary for cell to duplicate itself and so on to reproduce. In fact, DNA is such an abbreviation that even politicians know it, because every time there's an election, I hear these things saying that's the time when DNA will appear on the front page of the newspapers. They will say DNA corruption is in the DNA of the Congress, and the Congress will say communalism is in the DNA of the BJP. So everybody knows what DNA is, and therefore since you know what DNA is and you're able to abuse other people with the word DNA, you might as well look at all the other chemicals that follow in bringing us to the stage where we are today. In biology itself, there are three pillars: genetics and evolution, both of them erected in the 19th century, Mendel and Darwin, and chemistry is what connects genetics and evolution. Variation and natural selection is through mutation and selection, and that's chemical, because after all, what is the Watson-Crick DNA double helix? It's polynucleotide chains, hydrogen bonds between bases. We'll say AT, GC base pairs, 3 hydrogen bonds for GC, 2 hydrogen bonds for AT. It's all chemical structures. The fact that you don't write them down as chemical structures doesn't take away from the fact that it's all chemistry. The hydrogen bond is a chemical construct. The question on what is life has been asked earlier. It was asked by Erwin Schrödinger, the founding father of quantum mechanics. And in the 1940s, in a series of lectures in Dublin, he asked this question: what is life? He asked this question because at that time it was known that living organisms transmit characteristics to succeeding generations, and therefore there's a unit of information transfer, and that unit of information transfer is the gene. And Schrödinger was interested in asking the question, what is the gene? He argues that the gene must be a molecule, and if the gene is a molecule, what molecule is it? At the time he asked this question, Oswald Avery's work on DNA as the pneumococcal transforming principle had not yet appeared in the literature. 1943 was that paper. The series of lectures was around the same time. But the word gene itself appeared in 1911 in this paper by Johansen. And he says at that time that he introduces this word, and then he says the science of genetics is in a transition period, becoming an exact science, just as chemistry in the times of Lavoisier, who made the balance an indispensable element of chemical research. When Lavoisier introduced the balance and weighing into chemistry, he made it an exact science, because the one thing that the balance taught us was this. That's how he discovered oxygen, that is, matter can neither be created nor destroyed. So if something disappears from your flask when you heat it, it's gone into the gas phase. If you are able to weigh it, then match the weights, you'll find one form of matter has been converted into another. These are things that you have learnt a long time ago, but sometimes when you think about them in the context of the more complex organisms that you deal with, they do make some sense. Nothing's transformed chemistry as much as the balance. And nothing's transformed biology as much as the word gene. Everybody is taking genes from one place and putting it to another. And today, if you have the technology of CRISPR, Cas9 and have gene editing methods, you are editing genes all the time. Now, it turns out that you are able to edit genes faster than you can edit sentences. And one only hopes that you are doing it correctly and that it don't make the same kind of mistakes that one does while writing. That's Johansson there. I've proposed the terms gene and genotype and some further terms as phenotype and biotype to be used in the science of genetics. So it's Schrodinger had other questions. Schrodinger had questions about, does biology violate physics? Does it violate the second law of thermodynamics? Because a cell is a highly organized entity. Thermodynamics tells us that the entropy of the universe keeps on increasing, and therefore organization means you have to pay a cost, and therefore is there a violation in biology? Now that he talks about order, disorder, entropy and asks this question, but in the 1970s the biophysicist Morowitz analyzed the problem and put it somewhat clearly, although this was known in the literature by that time. He says, in a steady state system, the flow of energy through the system from a source to a sink will lead to at least one cycle in the system. Look at the word cycle here. The word cycle is very important, because if you take a biochemistry textbook, one of the most terrible things in biochemistry textbooks are all those cycles. Nothing turns a student of biochemistry more than seeing the Krebs cycle or the Calvin cycle. When you see the Krebs cycle or the Calvin cycle, you find that things seem to be going round and round and everything is in perpetual motion. But you have to look carefully at what's coming in. And you will find that anything that's coming in, some molecule which is there in the cycle has made one more molecule of that. So this is a wonderful way of biosynthesis. This is the chemical complexities of life. There are two charts which I like very much. One is the biochemistry, the metabolic chart, and the other one is the periodic table. When I was young, they used to hang on the walls of chemistry department lecture halls and biochemistry department lecture halls. And the wonderful thing about both the charts was that nobody ever looked at them. And you always wondered why were they there. Now I would urge you, because nowadays in the modern era, those charts have disappeared. I think those charts should be put back on, slightly maybe made more glamorous, and one should look at them, because when you look at them, you will raise many questions, and those questions are being raised now. For example, this chart, I would like to describe this as, it represents like a traffic map, like a road map in a city. This is the highways, crossroads, streets and bylanes of cellular chemistry. You will find that everywhere there is a diversion. If something, you can't have jams here. You can't be like Bangalore having a jam everywhere, because if you do, you have disease. And therefore, almost everything that we call metabolic disease is because something's gone wrong in this complex picture. But if you're thinking about evolution, you can ask the question, how did all of this come about? And if you ask that question, you ask, what is the logic of this kind of complex network of chemical reactions? That is the question which is now being asked, to ask how does energy get created, how does that energy get distributed, and the cell is extraordinarily sustainable. It's doing two processes which you all know and don't like to think about. One is it's making molecules, the other is it's breaking molecules. So on one hand it will break molecules like when you say fatty acid oxidation. In another case you will say it's doing biosynthesis. Catabolism, anabolism, these are the two components of metabolism. Chemistry is complicated, and biochemistry even more so, because there is so much of it. But I found this last year. I teach a class on biochemistry to the most reluctant students of biochemistry who are usually cell biology students. And cell biology students usually consider themselves very superior because they are using confocal microscopes where you see red and green and blue colors, you do not see molecules. And but this appeared in Nature 150 years ago in the year 1874, and you must read it. He says, the man who jokingly said that he had to give up the study of chemistry when the science became so bulky that its handbook required a wheelbarrow for its conveyance, expressed a truth which has been painfully felt by scientific workers. So, in 1874, they felt there was too much chemical knowledge, and so better not to study chemistry, better to study some other subject. And he says here, chemistry of all the sciences is perhaps the most unlimited capacity for development. Its subject is enormous, including the whole of nature, animate as well as inanimate. And this is true. After all, just now before I came into this lecture hall, your director took me and showed me a room where full of perfumes. And in each perfume, there it is, a bottle with some liquid inside it. That liquid is a chemical. In fact, I find aldehyde written on top of it. So, useful to know what aldehydes are if they are going to be molecules that you can sell for $10,000 a gram and so on. So, molecules are generally useful. Drugs are molecules. What was known in 1874? I asked myself the question in preparing for this lecture, and this slide is a new one. And 1874, although nature is complaining that there is too much chemistry, what was known in 1874? I have put those things which are known there. Every one of you will know these, and therefore there is no excuse for saying that I have not heard of this before. The first one there in 1828 is Woller's synthesis of urea. And so all he did was to take ammonium cyanate and heat it, and he got urea. He got so excited when he produced urea by heating ammonium cyanate. Ammonium cyanate was an inorganic chemical. He heated it, he got urea. Urea was something which comes from a kidney, so they already knew that urea was biological. And then he wrote to Berzelius saying that, very excited, he says that I have made urea without the use of man or dog. And then he can say, I am so excited, he says, I can no longer hold my chemical water. And he had to write the letter. That was 1828. 1848 was when Pasteur resolved tartaric acid and introduced the idea of three-dimensionality in molecules and chirality. And 1865 was when Kekulé had a dream and had the snakes catching their own tail and wrote a cyclic structure for benzene. And the last one is 1874. That is when Van 't Hoff and Labelle gave the idea of carbon having a tetrahedral valency distribution. And molecules went into 3 dimensions. This was before our understanding of atomic structure and atomic physics in the way it is today. Even then, chemistry was very vast, because chemistry, you can take anything and mix it together and get something else, and you can get lots and lots of substances. But nature also makes substances. You call it metabolites. Plants are making useful metabolites. You hope to make money with plants. How do plants make all these remarkable metabolites, terrible metabolites, chemically very complex? They make them because you convert molecule into molecule into molecule, substrate into product, product becomes substrate, next reaction, next reaction, next reaction. Each one of these requires an enzyme, which is a protein, and therefore each one of those requires a gene. This is what you will now clone genes, and you would say metabolic engineering. But you must ask the question, in order to make this small molecule, the plant is investing a huge amount of energy. It is making proteins, it is making genes, and then all of them are used to make this molecule. Why is it making the molecule? Of what use is it to the plant? The plant does not want you to feel happy smelling the aroma. That is not biology. The chemical complexities are such, vitamin B12, which is essential for every organism, because the enzymatic reactions involving vitamin B12 cannot be done in any other way. Vitamin B12 is so complicated, 36 enzymes are needed to produce this, which means 36 genes and all that control. This is the most abundant protein on the planet, rubisco plant. This is what is harvesting light and fixing carbon dioxide and producing carbohydrate. It is so inefficient as an enzyme that you need a lot of plants to do a lot of photosynthesis. And can you improve this? So, a paper just appeared earlier this year with a huge amount of work. They come to the conclusion that it is probably not possible to make rubisco more efficient than what it is. Nature has somehow found a way that rubisco is rubisco and is doing its own thing at its own rate. You can't actually make this go better. And he says, careful balance, its apparent inefficiency is a careful balance of several biochemical trade-offs between rate, affinity and promiscuity. So, one has to read between the lines. I won't, that's quick. Francis Crick, as early as 1958, when very little was known about how the genetic code was being put together, how was it now being read and so on, he actually said this, that at first sight paradoxical, that it's probably easier for an organism to produce a new protein than to produce a new small molecule. And this is true. Metabolites are tough to make. I like this quotation about biochemistry, which is the subject that I try to preach about, written by Sidney Brenner. He was an acerbic critic of many things in science. He said that I once made the remark that two things disappeared in 1990. That's when molecular biology was beginning to reach its peak. He said one was communism and the other was biochemistry. That was when the Soviet Union collapsed, and that only one of these should be allowed to come back. Now, it turns out that Brenner hadn't come to Bengal at that time, and Brenner hadn't come to Kerala even today. Otherwise, he would have known that communism still sort of survives in pockets. And then, of course, the Soviet Union itself is likely to come back the way things are going. But he says about biochemistry that we don't have to resurrect biochemistry because it remains the only experimental basis for the causal understanding of biochemical mechanisms. Historically, if you look at the evolution of biochemistry, this is how it went. Began with microbiology. When infectious disease, Pasteur, Koch said that microorganisms cause disease. What is the basis of that disease? That started off much of biochemistry. But if you go further, nutritional disease, that led to the isolation of the vitamins. And wow, what are the vitamins doing? They're all coenzymes. That led to the enzyme hunters. And then when cloning came, it led to what I would call the gene hunters. Now, of course, after the genomes have come in 2000, 25 years have gone. I don't have another horizontal line there because history requires that you step back, and hindsight only allows you to decide what is a historically important term. But if any of you have any idea of what I should put on that question mark, please send me an email and I'll look at it. You need some elements for life. You want to make life, you need some elements. Elements are very important. The periodic table is something that is going to determine much of what happens over the next few years, because I know that most students, and I've taken a poll almost in every audience that I've given a lecture, no student I've come across, modern student, reads a newspaper. And it also turns out that the overwhelming majority don't even look at news on their phone. So they generally are quite happy and they don't know what goes on in the world around them. So if you want to ask, why does Mr. Trump want to take Greenland or Canada, and why does Mr. Putin want to take Ukraine, the answer was clearly in the periodic table, because all they want is what is underneath. Now, nobody wants to take India, there's nothing underneath. Geology isn't on our side. But if, for example, you had some of the elements of Mendeleev's periodic table, which are important for technology, then of course everybody would be here. But these are the elements important for biology, and you should look at them a little bit. But the periodic table itself is something which people have forgotten. It also will important in asking another question: can there be life on any other planet? And if there is life on any other planet anywhere, will that have the same kind of biochemistry? Is this biochemistry universal? The physicists, of course, believe that physics is universal. And if physics is universal, chemistry must be universal. And if chemistry is universal, then biology must also probably be universal. So unlike the science fiction movies, we are unlikely to find life which is vastly different. And but this is the best in the literature that I have found in a book called Uncle Tungsten, written by the neurologist Oliver Sacks. He describes the periodic table by describing Mendeleev. And he describes Mendeleev as a barbaric, extravagant figure, romantic in his own way as the Byronic Humphrey Davy. But then he says something which I really like. He says, I spend hours now enchanted, totally absorbed, wandering, making discoveries in the enchanted garden of Mendeleev. So the periodic table, if I want to introduce it to NBRI, I must call it a garden. But Oliver Sacks has already called it the enchanted garden of Mendeleev. So this is where you see diversity of the elements, all of them of course controlled by atomic structure theory. The wonderful arrangement of these elements is such that carbon and silicon lie in the same row. And there it is. Carbon is what you need for biology and life. The digital information age requires silicon, two elements. They are driving your life today, and so it's useful to pay a tribute to Mendeleev every now and then. Where does all these elements come from? They are the Earth's inheritance when it was born 5.6 million years ago. You can't make elements on Earth. You use them up, you're finished. That's why the word sustainability becomes so important. You can't keep on mining and taking out the elements and not recycling them. It will catch up with you at some point or the other. We don't worry about it only because of this, because anything that is going to happen in the area of the environment is going to happen over a long time scale. And our time scale at best is 100 years. And we don't worry about it. But the Earth's time scale is on billions of years. And here, of course, I'll leave you with one thought. You know, life will come to an end. Biology will come to an end. And you can ask the question, when will it come to an end? That we know. Except it's somewhat far into the future, because the physicists already know, beginning all the way with Subramaniam Chandrasekhar's famous work, the stars die out. And when will the sun die out? The sun now has an estimated life of another 5 billion years. So we already finished half the time. We finished half the time learning all of whatever we've learned, and then we've got a little less than that to go. But as the star begins to die out of energy, all things will happen to around it. So life as we know it has a finite time. And it's only the stars where the elements can be made. So when the Earth and the solar system were formed, that's the time when the Earth got all its elements. He says matter itself evolves in the stars. And then he says the word comes from Darwin and biology, but it is the word that changed physics in my lifetime. And biology, respect for biology must be there from the fact that how is carbon formed? Without carbon, there is no life. And carbon is formed in a most improbable way in the stars. And all the carbon we have on Earth has been formed that way. So life is extremely improbable. And it's very improbable that you will find it in another planet. You have to look at a large number of planets. Fortunately, the universe is very, very large, and therefore there's every possibility at some time in the future that it would be there. Darwin himself thought about this, but I won't spend time on it. Haldane then worried about this later on. And there have been experiments which have done, but I'll skip over them, but come back to the timetable of evolution. And when you come to the timetable of evolution, the transformation really is around the time of the invention of photosynthesis. So it is here that the plants become very, very important. Eukaryogenesis, the first eukaryotic cell, appears around that time, at the beginning of the oxygenation events. The eukaryotic cells come by fusion of an archaebacterium and a bacterium. One of them then becomes the mitochondria. And this idea appeared in this paper in 1967, what is called the endosymbiotic origin of the eukaryotic cell by Lynn Margulis. But as far as biochemistry is concerned, look at the structures that I have written down there. Even the oldest of organisms that we can study has the same genetic code. The oldest of organisms that we study has the same way of producing energy, the electron transport chain, and a different electron acceptor, not oxygen, different electron acceptor, but the same thing. Strip hydrogens and atoms, electrons and protons, electrons going down the electron transport chain, protons going to one side of the membrane, potential built up, and this used for the synthesis of ATP. So, ATP synthesis, genetic code, common to all living organisms. So, all these kinds, cycles, the electron transport chain, molecules like vitamin B12, heme, porphyrin, heme and chlorophyll, all of that must have come before events. You can't invent photosynthesis without inventing chlorophyll. So, you must have an abiotic synthesis of chlorophyll or an abiotic synthesis of heme. But after oxygen has come, here are the molecules, and this is what I would really like you to appreciate: cholesterol, lignin and collagen. I have taken 3 molecules. Cholesterol, because it is the beginning of the steroids, and that oxygen atom in cholesterol comes from molecular oxygen. Cholesterol is the basis of the sex hormones. Cholesterol is the basis for controlling membrane fluidity. Cholesterol starts the synthesis of the bile salts. Cholesterol is important everywhere in human physiology. And lignin, without lignin, nothing that you see around in your museum or in your garden is possible, because in order to have photosynthetic efficiency, trees must grow big. You can't just have a cyanobacterial mat doing photosynthesis. And if you want to grow big, you must have structure. And if you need structure, you have. And then look at us, we need collagen, most abundant animal protein. If we don't have collagen, we don't have all this structure. And therefore, plants, animals require different ways of getting structure. Plants get lignin, animals get collagen. Collagen, proline gets hydroxylated in a post-translational modification. That requires molecular oxygen. Every one of these biosyntheses requires molecular oxygen. So, there you are, the very complex molecules. I will skip over them except to tell you that chemical complexity underlies given up. What happened to this? Huh? This one? Yeah. Oxygen is still the basis of cholesterol synthesis, and I want you to show you this. This is how cholesterol is biosynthesized. You see that oxygen atom right there, the second structure. A linear molecule squalene, which you will recognize, becomes that complex tetracyclic structure, and all of this in one step, and this is because of oxygen. Otherwise, we would not have animals if you did not have this kind of reaction. And this is what one would requires to make an essential molecule. There we are, tree of life, and what we are looking at is trying to understand how to put together LUCA, or the last universal common ancestor. Starting materials, very simple. This is all that we had to begin with. A little bit of extraterrestrial help from comets, where hydrogen cyanide and formaldehyde can come. They are there in comets. You can take hydrogen cyanide and you can make molecules, polymerize them, you begin to see the elements of the nucleic acid bases. This is abiotic synthesis. Is there a common chemical model for life in the universe? This is a question which people do ask nowadays. This is a 2004 article, and I like the quote here from Alice in Wonderland, because Alice tells the queen at one point, oh, that's impossible. But the queen says, why, sometimes I have believed as many as 6 impossible things before breakfast. and therefore, in imagining how the first cell might have formed, you need to use your imagination. Energy is what you need for life, and the cell produces energy with the electron transport chain. You take it for granted, but underlying this electron transport chain is Mitchell's idea of chemiosmosis and electron transport and how the chain is driven. Electron transport is important. In fact, one of the best quotations I've seen is by St. George, who says, life is nothing but an electron looking for a place to rest. And there's no more succinct definition of respiration than that. So, when you stop breathing, you stop producing, the electrons have no place to go. You've run out of oxygen. How did all of this come about? The iron sulfur clusters, minerals have the same kind of iron sulfur cluster. So, in early days before the first cell, Mitchellian electron transport could have been invented. But one thing I wanted to tell you, because it's a biology audience, the chemiosmosis concept, the chemiosmotic theory of Peter Mitchell, is something which fetched a Nobel Prize in 1978. But it's something that is glossed over in the textbooks completely, and nobody pays any attention to it anymore. But it also turns out that any assessment of biochemistry leads to this, and this is written by a theoretical chemist many years ago, who calls the idea of Mitchell as the most counterintuitive idea in biology since Darwin, and the only one to bear comparisons with the ideas of Einstein, Heisenberg and Schrödinger. And this is true, because in biology, for life you need to produce energy. Energy is a molecule, ATP. You need to produce that ATP. How do you produce that ATP? What energy are you tapping? It's the energy of the hydrogen atom. And the hydrogen atom is the simplest atom that you can think about. One proton, one electron. You have to take them apart. You push the electrons through the electron transport chain. You pump the protons to one side of the membrane. The moment you've done that, you have a membrane potential, you have a battery, you have energy stored, electrochemical energy. And now you convert the electrochemical energy to do the most thermodynamically unfavorable reaction, which is the phosphorylation of ADP to ATP. Now, of course, all of this has come about. Go back to Bronowski and his Ascent of Man. He says, among the multitude of animals which scamper, fly, burrow and swim around us, man is the only one who's not locked into his environment. His imagination, his reason, his emotional subtlety and toughness make it possible for him not to accept the environment but to change it. He says that series of inventions by which man from age to age has remade his environment is a different kind of evolution, not biological but cultural. I call that brilliant sequence of cultural peaks Ascent of Man. Why have I put this quotation here? Because it uses two terms here which you're going to hear a lot more about. One is biological evolution, the other is cultural evolution. You hear the word culture a lot more today. What is culture? If you look at Bronowski's book carefully, you will see that every cultural advance which is attributed in human civilizations has been driven by a scientific advance which happened just before that. Whether it is domestication of animals, whether it is the invention of agriculture, all of this as we move from hunter-gatherers all the way down to a modern city-state and civilization. So, science and technology are really underpinning the basis of human culture. Now, of course, you might ask, what is the difference between history and biology? This morning when I went around, your director showed me a tree. And he said the tree's been there for a long time. It was there during the first war of independence, so 1857, well, quite a long time ago. And the tree's been there, and what's the tree famous for? The tree is famous for the fact that it was seen where some historical incident happened, where a defender, for example, may have attacked offenders from the vantage point of a tree. There may be other trees in which people have been hanged. This is human history. We began today with a 2-minute silence for the victims of the terrorist attack. That is human history. This is what human beings are a part of biology, but They sometimes forget that they are single species. And it turns out you can ask the question, when did human history begin to differ from what I've been telling you and trying to put together, which is evolutionary history. That's the history of biology. That's the history of life. You can say the cognitive revolution, the sapiens, Yuval Harari, he says this, the cognitive revolution is accordingly the point when history declared its independence from biology. That's when the brain developed. And when the brain developed, one might ask the question, what does the brain now produce? Your brain now has imagination. It produces things. What does it produce? It produces stories. And Harari goes to say, no longer enough to comprehend the interactions of genes, hormones, organisms. It is necessary to take into account the interaction of ideas, images, and fantasies as well. So, very often we are driven by fantasy, we are driven by mythology, we are driven by things which are not based really on the kind of scientific fact with which we have looked at the study of. We have not understood how the cell was made. We have a long way to go, but at least in trying to understand how the cell was made, we are asking the question, how did every individual component come about and how might they have been put together. So, I hope that you will have the opportunity to think about some of these things, read about them, because I sometimes feel that I am running out of time, and it is quite unlikely that more enlightenment will really follow in the next few years. But in concluding, I must acknowledge only the environment in which I have been, the two institutions in which I worked, the Indian Institute of Science on the top left, where I spent my entire official professional life, 41 years, and the National Center for Biological Sciences in the bottom right, which has provided me a shelter in retirement. Thank you.
Yesterday we had a wonderful lecture, and today's lecture was icing on the cake, isn't it? It was such a beautiful lecture about life and everything, and many of us keep asking this question that it's all CH and how is it life being formed? Excellent.
Thank you, sir, for such a wonderful lecture. I'm sure for all of us it was a delight and honor to have you here. So I now request Director sir to please felicitate sir with a token of our appreciation. Now to deliver the vote of thanks, I invite Dr. Aditi Gupta, Senior Scientist, on the dais.
Thank you, Shweta. Good morning, everyone. It is a great privilege for me to deliver this vote of thanks on this special occasion where we are paying our tribute to Professor K.N. Kaul, an iconic figure in Indian plant sciences and the founding father of CSIR-NBRI. First and foremost, I extend our deepest gratitude to Padma Bhushan Professor P. Balaram, former Director IISc Bangalore, for gracing this event as our chief guest and delivering this truly thought-provoking and insightful lecture. Sir, we are deeply honored by your presence and inspired by your words. Thank you. I sincerely thank our director, Dr. A.K. Shashni, for his continued support, vision, and encouragement that make events like this possible. A special thank to Dr. Vidhu Sane for her insightful introduction of our esteemed speaker. Also, I would like to acknowledge the efforts of our administration, ICT, garden team for their seamless coordination behind the scene. I extend my heartfelt appreciation to the faculty members and all the students who have been working enthusiastically and taking participation in these activities these 2 days. Finally, I would like to also thank the personnels from media to join us here and cover the event. And my heartfelt thanks to each and every one to you in the audience for being here today and making this event a meaningful celebration of Professor Kaul's legacy. Thank you.
I now invite you all for high tea, which is being held outside, and for the dignitaries, high tea is being held at Gladiolus room. Thank you. After tea, we have next session for SPSF where we'll be talking with our alumni. So please be here within 10 to 15 minutes.